During normal breast tissue development and maintenance, cells grow and divide as they do in any tissue. And as we learned about in Lesson 6 with many other cell divisions, mutations occur all the time. Every time the DNA is copied to go into new daughter cells, there's an opportunity for new mutations to become introduced. Typically, most all of these mutations are repaired or fixed so that the cells can go on performing their normal functions and providing structure and function to an overall tissue in the breast. But if enough mutations in critical genes go unrepaired, then there's a high likelihood that cells will start growing out of control and form the beginning of a tumor that you can see here. Now remember, mutations are going on all the time. And so your body is really constantly fighting a losing struggle in some sense, because mutations will keep happening, and at some point, if your body isn't able to repair them all, one or two or possibly more are going to disrupt normal function and likely lead to the initiation of tumor formation. Now, obviously, there are many important proteins responsible for repairing mutations when they happen, but two that are really relevant for breast cancer are the genes called BRCA1 and BRCA2. We might call them BRCA1 and BRCA2. Carol has a mutation in BRCA2. Other women have mutations in BRCA1. Either way, mutations in either of these genes disrupt this process of mutation repair and can lead to a much higher breast cancer risk. Let's chat a bit more about BRCA1, which has been pretty well studied. The BRCA1 gene can be found on chromosome 17 in the human genome, and chromosome 17 has about 81 million base pairs, and BRCA1 can be found about halfway down the length of the chromosome. The gene itself has 23 exons spanning 80,000 base pairs of DNA. After transcription and splicing, the resulting mRNA has 5,592 nucleotides of coding region between the start and stop codons. This results in a protein that has 1,863 amino acids. Now, there are multiple alleles of BRCA1 that are associated with the high risk of early onset breast cancer, but one allele in particular has an insertion of a cytosine at position 5,382 of the coding region, as shown right here. Using the genetic code that we learned about in Lesson 3 and that you can find about in the instructor's notes on this page, I want you to determine the expected change in amino acid sequence and then select the type of mutation that you think this is best categorized as. Again, if you have trouble answering this, review mutations from Lesson 6.